as well. Welcome back. I hope everyone had a good reading week. No? I hope you weren't sleeping on the streets too much. It's been an interesting time, interesting few couple of weeks. But anyway, it's a little bit of we've had a little bit of a disruption with reading week and the continuity of the class. But actually, reading week provides a nice break. It's a nice transition in this course from doing a lot of the intensive laboratory stuff to start thinking about things a little bit more deeply, learning a little bit about advanced analytical techniques and some of the things that you might not experience in your coursework so far. So the rest of this semester is going to rely on some lecturing, some demonstrations, more demonstrations that we had in the past of high-end equipment that we just don't have enough of them for everyone to learn how to use them one by one. But we will try to demo some of this equipment for you. And also we will do some more field activities and lab activities so you can get some hands-on experience. So where have I been? Well, I went home. I went back to the U.S. primarily to visit my family because I hadn't seen them in over a year. But I also used the opportunity to visit some interesting American higher education institutions. I went to Penn State University to visit with a colleague and talk about research. And then I went to Princeton in New Jersey. It was my first time to both of these universities. And both of them have very beautiful campuses. Princeton especially was quite a nice place to visit. But Princeton I had some big challenges, I had a lot of homework to do, let's say. I was invited to give a talk in a seminar series called EGGS, Environmental Geology and Geochemistry. So this is a group of people who are more geotypes, but notice it says environmental, just like we're studying environmental science. They're interested in the geosphere, but they're also interested in the interactions between the geosphere and the biosphere, so the living world as well as the non-living world. So you can see I was indeed there. There's the evidence of it. And it was an interesting experience, quite an interesting place, a lot of history there. But in addition to the seminar I gave, we were collaborating with a research group there and doing some environmental science. So why Princeton? Well, at Princeton there's a group in the Sigmund lab, Danny Sigmund's lab, which is doing some really advanced stable isotope analyses. And the reason I bring this up is because today's lecture is on stable isotope analysis, which you will find is a really incredible tool for studying environmental science. So in this image here, this is my postdoc, Nicholas, and my master student, Phil. They are drilling a coral core. So this big lump that you can see here is a massive, we call it a massive Porites coral. It's a big hunk of limestone that has been secreted by the animal, which is just that thin surface layer of tissue on the top of the coral. And what Nicholas and Phil are doing is they're using a drill that is being powered by compressed air to drill through that really dense calcium carbonate material that has been laid down by that coral ever since it was a little baby coral, right? So now it's a very big adult coral, and we can go all the way back in time to see what kind of environmental change this coral has experienced. So on this panel here, you can see an X-ray. We took an X-ray of this coral core actually at Ocean Park, because Ocean Park has an X-ray machine that you can fit a panda into. So putting a coral core in their X-ray machine is rather easy. So we can get this X-ray, and it may not be that clear to you from back in the room, but you can see that in the X-ray there are these bands. The bands are indicative of the seasons during which the calcium carbonate was created by the coral. So when you have a really dark band, that means you have a very high density layer of calcium carbonate that was created during the winter, because during the winter the coral is growing very slowly because it's cold. But then in the summertime it warms up and the coral starts to grow very rapidly, and then we get a band of very low density calcium carbonate. So every time we see a pair of a dark band and a light band, we know that that represents 1 year of time. So now that should get your wheels turning. because we can do time series reconstructions of past environmental conditions based on this coral core. It's very similar to doing dendrochronology. Dendrochronology is the study of the rings of things, right? So this is particularly relevant for trees and tree ring analysis. So we're doing essentially the analysis of tree rings, but in corals instead. Now, why Princeton? Well, Princeton, in this lab we were visiting, they have mastered a technique for analyzing the nitrogen isotope ratios of the skeleton. And if you know anything about limestone, you would realize that limestone, which is calcium carbonate, has no nitrogen in it. Well, in a biogenic carbonate, a carbonate that has been generated from a living organism, there is a little bit of nitrogen, because in order for that calcification to proceed, there has to be a framework of organic material that calcium carbonate can be deposited on and crystallized. So that framework, that organic material, represents about 0.015% of the mass of the coral skeleton. So we're talking about a trivial amount of organic material inside that calcium carbonate structure. And this lab that we have been working with, they have the technology, they have the methodology to analyze this tiny, tiny amount of stuff and get a nitrogen isotope value. Now, by the end of this talk, I hope you will understand a little bit more about what I'm talking about. I'm showing you the data that my postdoc Nicholas collected just last week, and we're all very excited about it because what you can see is that the numbers are increasing. Okay? They're increasing. So what does that mean? This is a coral that was cored in Guam. Guam is a very tiny island in the middle of the Pacific, which is a territory of the United States. It has a very troubled history with respect to World War II. So during World War II, the Japanese occupied Guam and many other Pacific islands, and it changed dramatically the population on Guam. In fact, at one point in time, the U.S. had bombed Guam so much that there were no living plants on the island. Can you imagine? It was just scorched earth. So now, today, there are no native plant species on Guam. They're all invasive species from other places. It's a really amazing history in this place. And when we drilled our core, we found out that we went back pretty far to 1958, but not quite to World War II, which was our goal. We wanted to see how all of that conflict affected the marine environment. But nevertheless, we can see what happened in the coastal ecosystems of Guam since 1958, and that story has to do with human development and pollution. So to get at those answers and to understand what those data mean, we have to delve into this subject of stable isotope ecology. And I use the term ecology because most of what we're going to be talking about, the applications of environmental science, are using organisms and ecosystems to understand the sources of certain elements within those ecosystems. So first, let's talk about terminologies. What is a stable isotope? Well, an isotope itself, if you break down the word, iso means same, and tope means place. So an isotope of an element means that it's occupying the same space in the periodic table of elements. Okay? So an isotope can be a variant. You can think of it as a different flavor of an element, right? It's functionally the same. It's functionally the same as any other, as a different isotope of the same element. And I'll give you some examples in a moment. The classic example is we hear the word isotope, many people immediately think of radioactivity. So the classic radioisotope example is carbon-14. Does anyone know what carbon-14 is used for? Carbon dating, radiocarbon dating. That's right. So carbon-14 is a radioisotope. By radioisotope, I mean it's radioactive. So carbon-14 has some extra neutrons, and it's unstable, and it loses those neutrons over time. That's the radioactive decay, and it gives off some gamma radiation in the process, which is not healthy for anyone. But radiocarbon surely has been used in science very productively to learn more about how the world works. The stable isotopes, on the other hand, they're stable. They don't change. They're born that way and they stay that way forever. Okay, so the stable isotopes that we're talking about today, just like all elements, are born in stars, and we'll talk about that now. You may have heard the term that we're all made of stars, and this is a really amazing and true concept. We're all made of star stuff, and we're made of, more specifically, of all the isotopes of the elements that comprise our bodies. These were all originating in stars. So there are lots of different examples, and when we talk about stable isotope ecology, we're mostly talking about light elements, not heavy elements, right? So the light elements in this figure, this is from a book and from a paper by a classic isotope geochemist named Itaru Wada, who works in Japan. And so this is the isotope person, and kind of a funny little thing. But you can see here, it's a depiction of a human being and the composition of a human being isotopically with respect to several elements. Carbon, you can see the head of the person is carbon with 2 C's. We have 3 oxygen isotopes that make up the body. And then one of the legs is nitrogen and the other leg is deuterium or hydrogen. So these are what we would consider to be all light isotopes. You could also throw sulfur in there if you wanted to, but that's not included. Now, we can contrast that with heavy isotopes. There are heavy isotopes of calcium, of magnesium, of strontium. So those heavy isotopes are measured differently, and they have different applications. We're not going to be talking about them today. But if we look at just the light isotopes, we can see that overall, the heavy isotopes, the rare isotopes that have an extra neutron, but they are stable with that extra neutron, those heavy isotopes are within our bodies, they're within all matter on the planet, but usually in a much smaller proportion than the more abundant isotope, the lighter isotopes that we have out there. So in the case of nitrogen, about 1.3 kilos of our body is the light isotope of nitrogen, but about 5 grams, only about 5 grams is the heavy stuff, okay? So these isotopes are within us, and they're within all matter that contains these elements. Now let's get into the terminology, and when I say heavy and light, what am I talking about? Well, I briefly mentioned neutrons, right? So let's imagine the periodic table in our minds, and we can zoom in on the element nitrogen. Nitrogen has an atomic mass of 14. So who can tell me what the atomic mass is a sum of? Protons and neutrons. Yes. So within an atom of an element, you have protons, neutrons, and electrons. Protons and neutrons have mass, and we give them a very arbitrary mass value of 1 atomic mass unit, right? So within nitrogen, we have 7 protons, 7 neutrons. And the charge of the protons is balanced by 7 electrons. But those 7 electrons that are whizzing around the nucleus they have some mass, but in relation to the nucleus, they have virtually no mass at all. Okay, so the mass of nitrogen, the atomic mass, is 14. Now, if we add a neutron to the atom, we actually don't change the charge of the atom, right? Because the neutron is neutral. It has no charge at all. We don't add any electrons to balance that charge because there's no change. But we do change the mass of that element quite dramatically, right? So the difference in mass between a nitrogen 14 and a nitrogen 15 is quite large. I mean, it's more than 5%, right? If we look at other elements like hydrogen, hydrogen has an atomic mass of what? Anyone know? Hydrogen is our lightest element. Well, let's think about that really hard. I'm hearing different numbers, so someone say something with confidence. Atomic mass of hydrogen. 1. 1 proton, 1 electron, right? If we add a neutron to that, it has no impact except changing the mass, but it's changing the mass by 100%, right? So isotopically speaking, the difference between hydrogen and its heavy isotope, which is called deuterium, is huge. And the implications for that in biology are also quite large. So what's one more neutron, more or less? It has a lot to do with mass and also abundance. So this is really the key to how we can use these isotopes to tell us about the flow of matter in ecosystems. So let's imagine Earth, for example. On Earth, we have 2 types of stable isotopes of nitrogen. And I used this cartoon or this photograph of these 2 fat kids. One is slightly fatter than the other one, right? So the guy on the left is representing our 15N, right? This is our heavy isotope of nitrogen. And on the right, we have the 14N, the light isotope of nitrogen. So they differ in their mass, right? But they look the same. They're functionally the same. They do the same things in all types of biochemical reactions. They form proteins just the same. They form amino acids just the same. They function exactly the same at the natural abundance level. It just so happens that in addition to the mass difference, the abundance of the heavy isotope is less than 1% of all the nitrogen on our planet. And that was kind of reflected in the isotope person, right? There was a very small amount of mass of 15N in us than the 14N. So these 2 features of the isotopes, their mass and their abundance, are critically important and make it easy for us to identify them in nature. Now we measure these isotopes by looking at the relative proportions of the heavy to light in a sample. And we compare that to some rather arbitrary scale for a baseline comparison. So let's again stick with nitrogen as an example. So for nitrogen, we are measuring, the measurement we're making is of delta 15N. You're going to hear me say delta 15N a lot today. But delta 15N is referring to the stable isotope ratio within some material. And it's calculated by looking at by actually counting. We're using an instrument to count the number of the heavy atoms and the light atoms in a sample. And that is represented by this fraction at the top, so the 15N over 14N in a sample. And we're making that value relative to a standard. In the case of nitrogen, the standard that we are using is the biggest pool of nitrogen on our planet, N2 gas, right? So a lot of this, I'm moving around with my hands, is nitrogen. And so what we do is we give that N2 gas an arbitrary baseline value of 0. And the values, the units that we're using for these values is per mil, or parts per thousand. But per mil is the proper units to refer to. So if we're measuring delta 15N in units per mil relative to atmospheric air. So what that means is that when we set our baseline using atmospheric air to 0, that means all other material can either be more positive or more negative than atmospheric air. Okay? So keep that in mind as we start talking about some real-world examples. Okay, so the atmosphere is 0. And if everything looked just like the atmosphere isotopically, then we wouldn't have anything to measure, right? So at some point, chemistry and biology act upon the isotopes that are coming from the atmosphere and changing the relative abundance of the heavy and light isotope in materials that are being produced. And in this case, we're switching to a different isotope, to carbon. And we're going to talk about what's called a kinetic isotope effects, which is really a mechanism that leads to a process called fractionation. So if we could just read this cartoon, sometimes the extra neutron makes a difference, right? It makes a difference. The example that that we're often hear is, like, imagine if you're just a robot and your job is to carry suitcases up some stairs, right? So you carry the suitcases up to the top of the stairs, and you go back down and you collect more suitcases. Now, if you want to maximize your efficiency, well, let's pretend first that you have big suitcases and you have little suitcases. If you want to maximize your efficiency, and all it matters is that you get suitcases to the top of the stairs, if you want to maximize your efficiency and reduce your energy loss, you'll take the small suitcases up the stairs, right? And you'll leave the heavy ones behind. And you'll repeat that for as long as there are light suitcases at the bottom of the staircase. But once you run out of the little ones, you have to start carrying the big ones. And this analogy is very similar to what's happening in your bodies right now, right? When we talk about biological isotope fractionation. So just like this, if we have a substrate here that is comprised of heavy and light carbon, it requires some kind of energy to get up the hill, this threshold for any kind of chemical reaction. It could be the process of digesting your food, or it could be the process of making a sugar in a plant leaf. All of those chemical reactions have some sort of energy threshold that they have to reach. And at a very molecular level, this stair analogy is happening all the time. So as you can see in this cartoon, as the 13C, the heavy isotope, is struggling to get up the hill because it's heavy, the 12C is already up there. It's already reacted. So it's faster. It's easier to process. And therefore, as this reaction kind of continues through time, we accumulate the light isotope in our products. Okay, so let's think about plants. Plants are making sugar using sunlight. Okay, so they're going to, just because of the efficiency and the speed of the reaction with the light isotope, they're going to process more of that light isotope, and that light isotope is going to end up in the sugars in a far more abundance than the heavy isotope. And for that reason, plants tend to have very negative carbon isotope ratios. Is everyone following me so far? Making sense? It's a little bit of an abstract way to think about things. So this difference in the efficiency of the reaction based on the mass of these little atoms, it can be thought of as a fractionation process. And fractionation is a term that you'll often hear in isotope ecology, describing the mechanisms by which isotopes change from a product and reactant. So here we have a very simple example. So let's say we have some kind of mixture of isotopes, right? And through some process, some biochemical reaction, we can have fractionation, where part of that mixture is being routed into one product, and the other part of that mixture is being routed into another product. And as a result, the isotope ratios of those two products will differ. And at some point in time, those products will become sources for other biological reactions, and they can be remixed together, okay? So in the process of fractionation, we are separating our isotope ratios. We're diverging our isotope ratios from the mixture. And during the process of mixing, we're recombining them together, and we would reach then our original isotope ratio. There's another important feature about all this, and it's whether or not these types of reactions are happening in a box. Is this a closed system or an open system? And this is important for some of the later things we'll talk about, especially when it comes to plant photosynthesis and the resulting carbon isotopes of plants. So in a closed system, you can see up here, a closed system, we have an internal pool of some sort of substrate, right? The raw materials that we need to conduct some sort of chemical reaction. And if we want to use those raw materials to synthesize something within a closed space, that's all we have to work with. And if we really, really, really need that stuff, we're going to process all of it to 100% completion. Okay, so in the beginning, as we start working on that stuff, we're only going to carry the little suitcases up the stairs. But at the end, we're going to have to carry the heavy ones too. So once we've reacted with all of that substrate, all of the suitcases are at the top of the stairs, and the isotope ratio is going to be the exact same as it was when we started. Does that make sense to everybody? But when we first get started in a closed system, when I first start picking up those light suitcases, the ratios are going to be very different. So over time, things diverge and then they come back together in a closed system. Again, it's a little bit of an abstract way of thinking about it. In an open system, things behave quite differently. So we don't ever have to go to completion. Let's just imagine that around me there's just a constant stream of suitcases. They're just piling up to infinity. So every day, all day long, I can walk up the stairs carrying tiny suitcases. I never ever have to handle the big suitcases. This can happen. So as a result, the heavy suitcases, eventually they pile up so high they spill out into the environment and go on somewhere else to someone else's pile. That unused substrate is now very much enriched in the heavy isotope, right? Whereas at the top of the stairs where I've taken all the light suitcases, the isotopes have become much lower because they're a much lighter isotope ratio. So hopefully that suitcase analogy is working for you. Okay, so how does this all work in a real isotope system? So let's talk about nitrogen in the biosphere. And this is a classic image of a slice of the planet, right? We're looking at the biosphere. And as I told you, the baseline for nitrogen analyses is atmospheric air, which we set to an arbitrary value of 0, which in the top you can see N2 gas is equal to 0. And so we can see through all these different pools of materials, such as pollution sources, fossil fuel emissions, soil organic matter, the leaves of plants, the nitrogen that's coming down from the atmosphere as deposition, the nitrate and organic matter that are in our freshwater ecosystems, the different plants that fix nitrogen from the atmosphere and turn it into organic matter, human-derived fertilizers that we're synthesizing using chemistry and energy to draw nitrogen out of the atmosphere and make fertilizer. That's a huge source of a unique isotope ratio. And then we have dissolved nitrogen in the ocean as dissolved gas, as organic matter, and probably maybe the second largest pool of nitrogen perhaps would be deep ocean nitrate, which occasionally percolates up to the surface water and has big impacts on biology of the ocean. So we can spend a whole lecture on each of these compartments, and I could tell you why the isotope ratios of those compartments is what it is. All the fractionations, all the processes that affect these average values that are reported in this figure. But that's not the goal of this course. What you should take away from it today is that all over the planet we have variability. We have variability in the sources and sinks of things like nitrogen on our planet. And you can get from here an idea of the type of variation that we can see. And if we think about pollution in environmental science, what's unique about human sources of nitrogen is that they tend to be very depleted. They tend to have very anomalously negative isotope ratios. Okay? So what's coming out of that fossil fuel emission is going up into precipitation. There's a fractionation associated with combustion that leads to this negative isotope value. And then there's a further fractionation during precipitation. So as a cloud moves across the landscape, it's raining down the really heavy stuff first, and it gets lighter and lighter and lighter as it moves along. They call that Rayleigh distillation. But you get more and more depleted as things move through the atmosphere. So you can see the range for deposited ammonium and nitrate can be really, really negative. I mean, negative down to negative 18, negative 15. So this would be very unusual to find as a source of nitrogen in a biological ecosystem unless you're in a very human-impacted place. And it just so happens that we have more and more human impacts as time goes by. Here's a figure just showing you how much nitrogen there is in our biosphere and where it comes from. So in this figure, we're looking at nitrogen in teragrams, which is a lot of nitrogen, since from 1900 to 2000, so for 1 century, the nitrogen that's being put into our biosphere. And the blue line in the middle there is the amount of nitrogen that's being derived from the process of nitrogen fixation. Nitrogen fixation is a very important process for the Earth's productivity. None of us can survive without nitrogen, right? You have it in your protein, you have it in your enzymes, you have it in your DNA. We need to get nitrogen somehow. But when I breathe in all that nitrogen, absolutely none of it is going into my body and being used for my life, for living, right? It's inaccessible. It's totally inert and abiotic. I can't touch it. But a few special groups of organisms on our planet have unlocked the secret to fixing that nitrogen. Cyanobacteria, plants like legumes, fungi like arbuscular and ectomycorrhizal fungi, if you've heard those words before. These are mostly symbiotic organisms that they, through evolution, came up with an incredible enzyme that can actually rip apart the triple bond of nitrogen gas and turn it into an organic nitrogen. And that forms the foundation for everything, all life on our planet. Everything that we eat, everything that we consume is either derived from nitrogen that's been stripped out of the atmosphere, either by natural forces or by human forces. And the human forces are evident here as well. So natural fixation is about 100 teragrams per year. Every year, 100 teragrams of nitrogen is fixed by nitrogen-fixing organisms. But starting at about 1940, when humans figured out that nitrates were excellent to create explosives, there was a great push to be able to mass produce nitrates. So the Haber-Bosch process was invented, which allows us to use pressure and energy to convert inert nitrogen sources into ammonium, into nitrates, into urea. And the primary driver of that was World War I and the need to find artificial sources of nitrates to create explosives. Prior to that, the sources of nitrogen for explosives typically were synthesized from guano, like the feces, the excrement of birds and bats and things like that. People were mining this stuff from natural sources to make explosives. But with Haber-Bosch, we didn't need to do that anymore. And that process, when applied to agriculture, which was the secondary application for making those nitrates and ammonias, when we applied that to agriculture, we saw the beginning of what we now call the Green Revolution. And the Green Revolution, which was backed by the synthesis of these fertilizers, is the reason for probably 80% of us in the room being here. Without this process, you would not be here, okay? So the only way that we can boost our agricultural productivity to feed everyone in this room is by using these synthetic fertilizers. And as you can see, the production of those fertilizers has increased almost exponentially, and it's been the primary driver for a huge increase in the amount of reactive nitrogen generated by human activities being released into the biological world. So today, we're much higher than where this figure ends. I need to update this. And we're starting to see the consequences of all this nitrogen in the biosphere. So here you can see in this gray window, between the 1960s and 1980s, we saw a period of global coastal eutrophication. The oceans, our lakes, our streams, they've all changed because of all this nitrogen leaching into the system and fueling productivity. So what kind of impacts has that had on our environment? And how can we trace that using stable isotopes? So in my lab, one of the things that we do routinely is use nitrogen isotopes to tell us about the dominant sources of nitrogen within the marine ecosystem. Okay, so there are a lot of different sources in the marine environment, and it can get quite overwhelming if you focus on those sources too much and think of them in absolute terms. A few of natural sources we have include fixation. We talked about biological fixation, 0 per mil. We can have deep watered nitrate upwelling to the surface. That has a higher isotope ratio, around 4 to 6. And then still higher than that, we can have natural denitrification. When a bacteria returns that nitrogen back to the atmosphere and gains some energy from that process, we can get a higher isotope ratio. And then we have 3 human-derived sources of nitrogen on this image. One is fertilizer. The Haber-Bosch process causes the fertilizer to be produced through chemistry to be depleted relative to the atmosphere. Sometimes negative, sometimes slightly positive, but it has a very similar isotope ratio to atmospheric nitrogen, which is its source. If we have atmospheric deposition, remember as that stuff moves through the atmosphere, it gets lighter and lighter, or sources of combustion tend to be very light. Deposition has a very negative value. That's from human-derived activities. And then sewage all the way at the top, just by the nature of how we treat our waste. We like to put all of our waste in one spot. We like to allow microbes to process those wastes rather aggressively. And in the process of all those chemical reactions, we're increasing the nitrogen isotope ratio through time. So the delta 15N of sewage can have an isotope ratio of greater than 10 per mil. And we're getting, at that level, we're getting to the limits of what would be natural in the world. Oh, it's really snoozing. Okay, so let's imagine we have a hypothetical benthic integrator in our ecosystem. This is a sea fan coral that grows commonly in the Caribbean. And this is an organism that I spent a lot of time working on extensively. So we can imagine a sea fan, if it's in an environment with all of these different sources, how are we going to tell what are the most important sources in that environment? It would be nearly impossible because all those sources are mixing. They're being mixed. And the coral itself is going to be integrating these different sources. So for example, if it's taking up nitrogen from fixation or upwelling, natural sources of nitrogen, it's going to reflect those sources. These sea fan corals are just like a plant. So they're absorbing that nitrogen just like a fertilizer in order to grow. So the coral would move to reflect those sources over time. And if we increase the amount of sewage in the environment, which has a very positive isotope ratio, then we would expect that the coral would move over time to reflect that increased sewage source. And similarly, if we had suddenly an influx of atmospheric deposition, we would get a change in the coral's isotope ratio to reflect an increase in that source. So sometimes we have to take a step back and not worry so quantitatively about these data, but rather use them in a more qualitative way to allow us to think about what sources are changing in the environment as opposed to which ones are dominating. And this is effective because everything is a weighted average. Everything is a weighted average. So a coral growing in the ocean, taking up multiple sources of nitrogen to grow and reproduce, it represents a weighted average of all those different sources. So let's imagine the coral is on a sliding scale here. So in the top example, we have a coral that has 2 sources of nitrogen. One looks like nitrogen fixation at about 0 per mil. And the other source is, let's say, sewage from a pollution. But as long as that pollution is relatively low, and in this example, the proportion of that source is only about 20% of the total nitrogen, then the coral is growing mostly on nitrogen from fixation sources. So it gets drawn down to reflect that source, right? So 80% coming from fixation, only 20% from sewage. We can calculate a delta 15N of our coral at about 2 per mil. So it's a weighted average of those 2 different sources. In another example, if we have a different environment that has a huge sewage impact, 90% of the nitrogen in the ecosystem is dominated by sewage, then that signal is going to pull our coral towards it as it's assimilating that resource to grow. So we could calculate under the same conditions a delta 15N of about 9 per mil. It looks very much like source number 2. And this is what we can do quite commonly. So now I'll show you some real data. So we know that, I showed you that figure already, that agricultural fertilizers have been a huge input globally. But when did we start seeing a shift in the isotope ratios of organisms in the biosphere? So agriculture has been a large historical and chronic input to the biosphere. And during my PhD, I worked on a project which utilized museum specimens of these corals that had been collected since the late 1800s or mid 1800s or so. So we basically had organisms from the Caribbean Sea that had been collected for over 150 years, 170 years or so. And we could take these samples from all over the Caribbean, and we could see that despite the fact that they had come from a variety of different places, different environments, different histories, there was a signal in all that noise. And we saw that the isotope ratios were gradually decreasing through time. And this was a really interesting result. And we found this trend in several species. And we believe that this trend is caused by widespread use of agricultural fertilizers in the Caribbean Sea. Many island nations were essentially clear-cut. All the forests were removed for industrial-scale agriculture at some point in time. And we can also look at other organisms. So this is the Florida manatee. This is a marine mammal. It's similar to the dugong that you would find in the Pacific. And the manatee is a coastal marine mammal that feeds primarily on plant material. And they tend to like to eat things like seagrasses and other submerged aquatic vegetation from freshwater environments. So I worked with a student who, again, went to a museum collection and sampled bones, bone collagen. So we're talking about the organic stuff upon which we grow our apatite, our calcium phosphate that makes up our bones. So this student went and found these manatee skeletons that had been collected since the 70s, took small samples from them by drilling out that material, extracting the bone collagen and measuring the delta 15N. And again, we saw this negative trend. So around Florida, during this period of time, there was something changing in the environment that these manatees existed. And because agricultural fertilizers are huge in Florida, and because the first plants that are going to utilize that fertilizer after it's flushed from an agricultural field are underwater. And these manatees are essentially a big mobile integrator of that pollution source, right? They're hoovering up all these plants and they're integrating them into their bodies. They are a weighted average of the nitrogen in the environment. And so we can see that impact through time. Now, if we think about the Caribbean as an example, and it's probably true for many of the island nations of the Indo-Pacific, land use is changing. You know, from the Caribbean, for many centuries, islands of the Caribbean were colonized mainly by the British, by the Spanish, and other countries. They were clear-cut, so all of the native vegetation was removed in favor of crops, and then those crops were shipped off to Europe mostly for their growing population sizes. But now, today, we have industrialized agriculture, we have a global economy. It's not as exploitative as it used to be, at least with these island nations. And many people have realized that you can make a hell of a lot more money by building a resort on this beautiful island than growing sugarcane for, pennies on the kilo. So now we have a land use shift. We're moving away from agriculture. We're moving towards urbanization in these tropical environments. And as a result, we're getting more and more wastewater inputs. And probably the poster child for this is Bermuda. Bermuda has one of the highest population densities in the world. I don't think it's as high as Hong Kong, but if you go to Bermuda, you can feel the density. There's no skyscrapers there, but there are homes packed in very tightly, and they build on anything that can be built upon, which is also very similar to Hong Kong. Bermuda is basically a limestone platform. It's basically an ancient coral reef that is emerged from the ocean, and people have colonized it, and for many years it was an important hub in the Atlantic for trade. Today, Bermuda is so densely populated on top of this limestone geology that the groundwater has become so contaminated with nitrogen from wastewater that it's unsuitable for drinking. So everyone in Bermuda who has a home has this strange-looking roof on the top of their house. They're usually like a concrete slab, and they're whitewashed. They're covered in this white paint that has certain properties about it that prevent the water from becoming contaminated with bacteria. So every time it rains, the water from the roof gets funneled down underneath the home into a container so that the people there can use it for drinking water, because they cannot drink their own groundwater. It's pretty scary, actually. So they're at the mercy of the weather. If they don't get any rain, they don't have water to drink. Now, what happens to that freshwater aquifer underneath of the island is slowly leaking out into the surrounding ocean. And it just so happens, if you look at this figure, the dark blue images are really, really deep water. I mean, it just goes to nothing. Or it goes to infinity. And so most of the south side of Bermuda is well flushed with clean, clear, beautiful ocean water. So it carries away, it dilutes the pollutants and carries them away from Bermuda. But here in the light blue is an incredible platform, an incredible lagoon that is like a fairyland. It's a magical place filled with corals and fish and all kinds of biodiversity. This area, unfortunately, is semi-enclosed, so there isn't a lot of exchange with the outer ocean. And so over time, slowly, the wastes from humans can leach out into this environment and accumulate and have dramatic impacts on the ecosystem. So I was asked to go to Bermuda and use my isotope tools to help them figure out the extent of this problem. And so we sampled these corals from inside harbors. So these are enclosed harbors with a lot of population density. And then along the outside of the island, we call that a nearshore environment. And then in the middle of the lagoon, and then the outer lagoon being well flushed with oceanic water. And here you can see the carbon and nitrogen isotopes of all the samples that we took in that survey. So if we look at the outer lagoon, the blue points, we have very low carbon and nitrogen isotope ratios. And the mid-lagoon sites are largely the same except for a few. And the nearshore sites and the harbor sites are crazy different. I mean, they're really, really different, especially the harbor sites. We have isotope values that are approaching 10 per mil. 10 per mil. And later on, we're going to talk about trophic fractionation, so you'll realize how crazy this number is for a coral. If you measure my isotope ratios, I have a delta 15N of 10 per mil, about 9 or 10 per mil. So in order for this coral to have the same isotope ratio, it would have to have the same diet as me, right? Eating beef and pork and rice and all that stuff. It's impossible. It's impossible unless the source of pollution is coming from a human. Okay? So these corals are very unusual and a sign of some environmental impact. What was really cool about working in Bermuda was that they had a lot of different sources of pollution. And just as a side story, I went to one site where my colleague was asking me if I could help him figure out if pollution was coming from a marina where they have people living on the boats, people living on their boats and just using the bathroom over the side of the boat. They were very concerned about it. So imagine you're with me in Bermuda and you're standing on a dock, like the dock's like this, and you're looking at the land, and behind me is the harbor. And over here is the marina where people are living on the boats and going to the bathroom in the water. And behind me is a giant cruise ship like a spaceship out of Star Wars sitting behind me. And they're also worried about these things leaking or dumping sewage right behind me. And then over here in front of me, just behind a wall, is a captive dolphin operation. We have 10 bottlenose dolphins that people pay to swim with. Okay? So they're asking me, can we tell if the people are going into the bathroom in the water, or can you tell me if the cruise ship is illegally dumping? And this is the data that we got. So in the marina, we had isotope ratios that were a little bit high. They're a little bit high. And over by the ocean, closer to where the cruise ship was, they're a little bit lower. So are the people in the marina polluting their environment? Well, probably a little bit. But there's a bigger problem that emerged. There was a small little opening where water from the dolphin enclosure could exchange with the reef. And when we sampled corals near that opening, we found they had a very high isotope ratio. So these data are collected along a swimming transect. We just swam, and every few meters we collected a coral at the same depth. So we saw that huge spike in the nitrogen isotopes of the corals because we are picking up a signal from dolphin waste. So the dolphins are eating a lot of fish every day. They're going to the bathroom in their enclosure, and that water is exchanging with the reef environment. And this is an unregulated source of pollution that we found. And we did some other investigations. We found another study that had documented high levels of mercury in the sediments around this area. And they were unsure what the source was. Well, the source is the dolphins, because dolphins bioaccumulate mercury and they excrete some of that mercury in their waste. And so that mercury is impacting the local environment. And we did some math to determine that the amount of mercury coming out of these dolphins, if this operation would be in the EU, in the European Union, it would require regulation. There would have to be some management of that mercury waste that they're not doing in Bermuda. So the isotopes can be very informative for identifying problems and also identifying other problems associated with sewage that you may not have been looking for. And I stuck this slide in here just to introduce the idea of trophic enrichment, but we'll talk about that a little bit later. Trophic enrichment is the process by which isotopes, the heavy isotopes, tend to accumulate in a consumer. And this is the reason that I told you earlier that I have an isotope ratio of 10 per mil. This is the reason that I don't have an isotope ratio of 0, like nitrogen fixation. Because I am the product, my biomass is derived from my diet. They say in isotope ecology that you are what you eat, plus or minus a few per mil. So this is true especially for nitrogen. So when I consume a protein and it goes into my stomach digests that protein. It breaks it apart into the amino acids. And sometimes I'm using those amino acids directly to build muscle or some other type of material. And sometimes I want to break down those amino acids and unlock their energy. So I create nitrogen waste in that process. And it seems, it's unclear, but it seems that in the process of that deamination, breaking down an amino acid, somehow that preferentially excretes the light isotope. Instead of retaining that in my body, we're getting rid of it because we're burning it as energy. And as a consequence, the heavy isotopes tend to accumulate in our bodies. The difference that we see between us and our food is on average about 3.5 per mil, 3.5 units per mil. So in this case, we have this yellowfin tuna. And yellowfin tuna get pretty big. They get up to 1 meter in the fork length. I'm not quite sure what the fork length is. Something like that on the tail? I'm not sure. But anyway, they can get big. And so if we sample yellowfin tuna throughout their development, from when they're a little tiny fish to when they're a really big fish, we can look at their nitrogen isotope ratios and we can look for changes. And we see a change at about 40 centimeters of fork length. We see a trophic enrichment. So suddenly, the isotope ratios were around 6 per mil, and they jump up to 8 to 10 per mil. And that enrichment is associated with a shift in their diet. Suddenly, they got to a size where they were big enough to eat something that's slightly higher on the food chain. And as a result, the whole isotope ratios shift along with them. So this is a trophic enrichment factor, and we'll talk about this a little bit more in the future. But one way that scientists really get a grip on these types of dynamics is by captive feeding studies. There's several students in the ecology and biodiversity group who are working on things like this, looking at trophic enrichments, food web dynamics, and things of that nature. So here's an example from someone I know, Sora Kim, who did a project looking at sharks and shark isotope fractionation through their development. So she could get sharks as embryos, hatch them, and then she could feed them whatever she wanted, squid or fish, different food sources that have different isotope ratios, and then she could see how those isotopes were assimilated, fractionated within the different organ systems of the sharks, really laying down the foundational scientific basis for how these things work in nature. And these types of data are very informative for those of us who are using shark isotopes to learn something about environmental health. So that type of work is critically important. Okay, so let's see. We are about halfway finished. Let's take a break. So why don't we reconvene here in 10 minutes? Those of you who are snoozing can splash some water on your face, and we'll finish up when you come back. So let's meet back here in less than 10 minutes, okay? So just before the minute hand gets to the 10.
Okay. Oh, great. We're recording again. All right, so let's move on. And one of the last examples for nitrogen I'll give you is really crazy. And this was a really interesting paper that came out in a Nature's online journal called Scientific Reports that came out a couple years ago. And basically the conclusion is that palm oil, the production of palm oil, which is an increasing concern in environmental science, is causing big shifts in ecosystems, in the community composition of ecosystems. So the title says, Tim Tams fatten people and starve mantas. I put that on there because I was disheartened to learn that Tim Tams, which I think are delicious, are made with palm oil. And palm oil, if you're not aware, is a very cheap type of oil that can be produced from palm trees. So it can be produced in the tropics at very little expense, and it's a cheap ingredient to use in our food. And unfortunately, palm plantations have devastating impacts on environments. They're really, really bad. So a lot of people were upset to learn that Tim Tams are using palm oil. And this is an example of a study that looked at the effect of palm plantations, not only on terrestrial ecosystems, but on the marine environment. And so I'll take you through this as best as I can. In this example, we have forests. And in parts of the tropics, these forests consist of some diverse assemblage of tree species. And seabirds, which are out in the ocean foraging and eating fish, come back to nest in these native forests to raise their young, and all the while they're doing their business on the ground, right? And their business leaches into the soil, it fertilizes the trees, it gives them a source of nitrogen, and what's not used can slowly leach into the marine environment, where it can be utilized by all types of organisms. The problem is that when the trees are clear-cut and they're replaced by a palm plantation, the palm trees are not very desirable by the seabirds. They can't use them to nest. So the seabirds don't visit that location, and therefore there's less seabird waste going into the soil, and there's less nitrogen to drive productivity downstream from the seabird. And the data that they have here demonstrate the impacts. So you can look at each of these figures at the top, and they're measuring different things. And the red bars are always from the native forests, and the blue bars are always from the palm forest. So right off the bat, you can see that the native forests always have more of every variable than palm forests. The first one is the abundance of native trees. That makes sense. There's more in native forests than in palm plantations. Therefore, there's more birds in the native forests. Where there's more birds, there's more nitrogen in the soil from their waste. Where there's more nitrogen in the soil, there's more nitrogen in the plants. They have more nitrogen in their leaves. There's more nitrogen in the runoff from the land. And that runoff, this is where it gets really weird, the runoff causes the local ocean environment to have slightly elevated nitrogen concentrations, which leads to the production of more zooplankton, planktonic animals, that are the food for manta rays, giant manta rays that come and feed on them. So this one simple change to the native forest led to all these cascading impacts that altered the abundance of a huge marine animal visiting the coastline of these environments. And so what the authors conclude is that, people have to really decide what types of ecosystem services they want in their home island, right? You can have palm plantations, or you can have ecotourism, looking at manta rays, for example, but you probably can't have both. There is an isotope component to this study, and it's evident in these triangles here. And the triangle is standing for the capital delta symbol, or thinking about a difference, right? So we're always thinking about the difference here between the native environment relative to the palm tree environment. So with respect to soil nitrogen, the soil delta 15N is 6.2 per mil higher in the native forest than in the palm plantation. That's a huge difference. And that difference is driven by the fact that seabirds are top predators in the ocean. They're eating fish, they're feeding on dead animals, and their isotope ratios tend to be actually as high or higher than humans. They're basically eating the same kind of stuff that we do. As a result, the trees and the plants have higher isotope ratios in native forests than they do in the palm forests. Even intertidal clams, things that are living in the intertidal zone, have higher delta 15N values. They're benefiting from that bird waste. And when you get into the subtidal communities, you even see a difference in the filter feeding sponges. They're more enriched next to the native forests. And the zooplankton that are feeding on phytoplankton in that system also have a significantly higher delta 15N. So the isotopes are tracking the flow of the seabird waste all the way through this ecosystem and provide very nice complementary evidence to the other data that they provide. Okay, so let's talk about carbon isotopes for a moment, because carbon isotopes are really important in environmental science as well. And just like we had our examples before, here's just to remind you the different isotopes of carbon. So carbon-12 is the most abundant carbon we have on the planet. It's more than 98% of the carbon we have on Earth is the carbon-12, 6 protons, 6 neutrons. Carbon-13 is one of those stable isotopes. It's non-radioactive. It stays that way forever. And it has 1 extra neutron. And then we have our radioisotope there on the end, carbon-14. So carbon-14 is not stable, so it's not relevant to this study. But this is nice to take a look at to see the different atoms and their conformations. We're looking now at a similar figure to the nitrogen ecosystem diagram. So here we can see that the atmosphere is also a large pool of carbon. There's a fair amount of CO2 in our atmosphere, but it's incredibly small relative to nitrogen. Right now we're almost at 400 parts per million. And the CO2 in the atmosphere has a unique isotope signature of about -8 per mil. And this is important because CO2 is a nutrient for plants and all plant life. So that -8 per mil is going into terrestrial and aquatic plants, and they are fractionating it based on their chemistry. And we'll get into that in a bit. But again, just to give you a broad view of the different compartments of carbon on our planet, we have the terrestrial plant biosphere, we have aquatic plants and organic matter. You can see that when CO2 dissolves into water, we can get a different isotope ratio. So dissolved CO2 in the ocean is about 0. It's not -8. Dissolved CO2 in freshwaters can be also slightly more negative. And that has to do with the process of diffusion. across the surface layer of water, between the water and the atmosphere. So there's fractionation associated with that passive abiotic process of exchange. So you can see that plants in particular have an isotope ratio that's heavily fractionated from the source. Instead of -8, like CO2, plants become something like -28 or -27. So they are much more depleted than their source of carbon. And fossil fuels that we pull up from the deep earth that are derived from ancient plants have a very plant-like isotope ratio that can also be very depleted, -27 or even beyond that. I think we were just looking at a paper yesterday, natural gas from fossil fuels can have an isotope ratio of -40. So this is starting to push the boundary of what we would expect to find within a natural ecosystem on Earth. Oh, and I guess I should say that the nitrogen, remember, nitrogen gas is the most dominant source of nitrogen on our planet. Well, actually, carbon, the biggest pool of carbon is in the ocean. Carbonate and bicarbonate in our oceans, the carbonate system is the largest pool of carbon on Earth. And so we use carbonate as our baseline, our 0 point. So all the isotope ratios we're going to talk about today with respect to carbon are based on that baseline of carbonate, which is 0. Okay, so we know we've changed the carbon biogeochemistry of our planet. Just like with agricultural fertilizers, we are unlocking these reservoirs of deep stored fossil fuels, and we're basically combusting all of that depleted material and putting it back into the atmosphere. And this is causing all kinds of problems with global change. And one of the confirmatory studies to prove that global warming is associated with greenhouse gas emissions is revealed by looking at isotopes. And so what we call this is the 13C Seuss effect, because Seuss was literally Dr. Seuss was the guy who discovered this. And what this is that as a reflection of the changing source of carbon to our atmospheric CO2 pool. Fossil fuels tend to be more negative than current atmospheric isotopes, right? So our atmosphere today is about -8. Fossil fuels, as you saw, they're derived from plants. They tend to be -20 or more and more negative. So as we burn those fossil fuels and release them to the atmosphere, the weighted average of the atmosphere is slowly going to become more and more negative over time. And indeed, that's what's happening in a very regular and predictable pattern. It's shocking. So you can see that the isotope ratios have declined from just the late 1970s, from about -7.4 all the way down to about -8.4. And this has, so this type of effect will translate to everything on the planet. Everything that's based on photosynthesis is going to be affected by this change. And in fact, going back to my study looking at the nitrogen isotopes in corals, we saw the 13C Seuss effect in those samples as well. So we saw about a 1 per mil change in the corals over that history. And my study wasn't the only study to do that. Here is a figure that backs up what I just said. This change in the isotope signature of our CO2 in the atmosphere affects everything. So here is an image showing you the delta 13C of corals and sponges that are living in the marine environment. And somewhere in there is another line that's showing the Seuss effect, that 13C curve. And you can see they all pretty much line up all in the same line. They're all recording the same process, that 13C Seuss effect. So this is a really big impact on our biosphere, on global productivity. And it's a really powerful way of demonstrating human impacts on the biosphere. Okay, so getting into a little bit of the trophic ecology, the food web dynamics that we can gain from carbon isotopes, we have to talk about plants a little bit. And most of the plants on Earth have two different types of metabolisms. Some plants are C3 plants, or that are, we kind of consider this the norm. These are plants that are that have a very basic carbon metabolism that has evolved from a very long time ago. So they're simply taking up CO2 from the atmosphere directly into their intracellular spaces, and they're using rubisco, which is a very important photosynthetic enzyme, to conduct what we call the Calvin cycle, or converting CO2 into sugar. The C3 part of this, I think I have this correct, is that every time you make one turn in the Calvin cycle, you're fixing 3 carbons. Okay, so that's where the C3 comes from. C3 plants. Now, we also have C4 plants. C4 plants have a slightly different metabolism. C4 plants have evolved a mechanism for dealing with high temperatures, for dealing with life in the tropics. So C4, going back to C3 plants, a lot of C3 plants we encounter on our daily basis. Most types of trees, most of our leafy green vegetables that we eat are all C3 plants. Things that are C4 are grasses, palms. You can almost tell if anything is a C4 plant because it has leaves that tend to be long, and the venation of the leaves is parallel, right? Does that make sense to everybody? If you have some tong choy for dinner, you can see the veins in the leaf standing out like that, right? But if you go and you see a sugarcane plant, all of its veins are going in one way. The sugarcane is a C4, tong choy is a C3. Okay, and the different types of metabolisms they have a big impact on their isotope ratios. A C4 plant has to deal with water stress. They're growing in hot environments where evaporation can be make life very difficult. So in order to photosynthesize and obtain carbon, a plant has to have a constant supply of water. And some basic plant biology. Everyone knows what a stomata is? A stomata? Everyone know what that is? Some yeses and some nos. A stomata is a tiny little pore on the surface of the leaf, usually underneath the leaf of a plant, that can open and close based on the plant's physiology. The stomata needs to open so that CO2 can enter and the plant can conduct photosynthesis. Right? So the plant needs CO2 in order to make sugar. But the cost of doing photosynthesis is water. If something's going in, something has to come out. And what comes out is water. Okay? So if you are a plant, if I'm a C3 plant and I'm growing in a very temperate, cool environment, I can open my stomata all day long. It's not too hot. I can get plenty of CO2. Everybody's happy. But if I'm a grass or a palm tree, and I'm living on the edge of a really hot environment, I do not want to open up my stomata at all because I will lose precious water and I'll dry out. But then I have a conundrum. How am I going to take up CO2? How am I going to do that? So C4 plants have evolved a cool mechanism. They can actually separate the process of carbon fixation in space. So they can compartmentalize the area in which they are doing the Calvin cycle. I'll show you that in a second. But the output of all this is that C3 and C4 plants have very different isotope signatures. It's incredibly predictable. And it's based on the fractionations that the carbon is going through as it moves through the leaf towards the Calvin cycle. So in C3 plants, as I said, we are expressing the full fractionation that rubisco enzyme is capable of, something like -20 per mil. So rubisco is very picky. It only wants to use the light isotopes. to a tune of about minus 20 per mil. So we're starting off about minus 8, and we're ending up with minus 28, thanks to Rubisco. And so in a C3 plant, the system is operating like an open system. Remember that figure I showed you way back? It's like an open system. You have an infinite substrate pool of CO2 coming in, and therefore the plant can express the full fractionation and take only the light isotope and let the heavy stuff go away. But in a C4 plant, because they're compartmentalizing this process, they're closing off the area in which the Calvin cycle is occurring within their cells, they're creating a closed environment, a closed box. They're putting the Calvin cycle in a box. And as a result, they cannot express the full fractionation. So remember, the source of CO2 is about minus 8. So these guys are still fractionating a little bit, but not nearly as much. They have an average carbon isotope ratio of minus 13. Okay? So big differences coming from C3 and C4 plants. And this is kind of the basic model that we're talking about. Air and CO2 are going inside of the internal CO2 pool of the leaf. There is some CO2 diffusion back out. So if I don't want to use the heavy stuff, I can let it leak back out. And then eventually there's some CO2 leaked, fixed and stored in the plant. So this is a very simplified model of what I'm talking about. But in reality, this is what it looks like. So at the very top, you can see the CO2 molecule entering through a stomata. In a C3 plant, it just enters into the interstitial space. It can be conducted, it dissolves in water, can be interconverted as bicarbonate, transported across the cell membranes, and then there it is experiencing, it's interacting with Rubisco and becoming a sugar. And every cell is the same. We have these mesophyll cells, they're all the same. They're all doing the same thing. But in a C4 plant, you can see we have a mesophyll cell and we have a bundle sheath cell. They're two different types of cells. And the bundle sheath cell is that box that we're putting the Calvin cycle in. Okay, we're putting Rubisco in the box. So what happens is that when conditions are good, the stomata can open. I have plenty of water. It's not too hot today. Open up my stomata. I'm going to suck up a bunch of CO2. But instead of fixing that CO2 into sugar right away with my Rubisco, I'm going to lock it away as another organic compound. And we call that PEP carboxylase, or PEP-C. So I've created a different substrate from that CO2 source. And then, regardless of whether my stomata are open and closed, let's imagine the temperature gets really hot. I close my stomata. And let's say it's hot for days or weeks. I keep my stomata closed. But I have a lot of that PEP-C stored up in my mesophyll cells. I can transport it into my bundle sheath cell, where Rubisco is waiting to utilize the carbon that can be released from that PEP-C and fixed as a sugar. So this is a very efficient and smart way of dealing with water loss. Okay? But it has big and important isotope consequences. So let's look at some examples of how we can use these things to learn about the environment. Here is data from an interesting study where they were looking at soils from a push core. Remember we did the soil sampling the other day? Well, this is a study that took push cores from an environment where they could go very far back in time. So you can see the scale, the age is 750 years before present, 3,000 years before present. So this whole scale is probably going beyond 10,000 years. Okay? And what they can do is they can look at the carbon isotope ratios of the organic matter in the soil, and they can infer what type of plant communities used to exist there 10,000 years ago. That's a pretty cool thing that you can do. So what they found was that, of course, we know that grasses, remember, if you think about it very simply, they are a C4 plant, right? They have that parallel venation. So they're going to have a delta 13C of minus 13 per mil, closer to this side of the graph. But forests are mostly going to be comprised of C4 plants. They're going to have a delta 13C of minus 27 down towards this side of the graph. And you can see that all throughout time, the soil organic matter is a weighted average of those two sources, right? It's never just one or just the other, somewhere in between. But you can also see that the delta 13C of that organic matter changes through time, right? So at some point, around 500 to 700 years before present, some of these environments were more like grasslands. And then they shifted very quickly to forest communities through some sort of successional process. So we can use isotopes to look into deep time and see how things have changed. We can also use carbon isotopes to tell us about the source of carbon in food webs. So unlike with nitrogen, carbon is essentially conserved as you move up a food chain. I don't necessarily retain the heavy isotope in my body as I would with nitrogen. So there isn't that same trophic enrichment. Instead, carbon is kind of a fingerprint of the carbon that you're assimilating. So in this case, we have a seagrass habitat. And seagrass, as you will remember, is a C4 plant, right? It's a grass. But it is living in an aquatic environment, so there is an additional fractionation associated with the CO2 that's reaching it. So they tend to have isotope ratios around -10. In the oceans, another group of organisms like phytoplankton will have a delta 13C of about -20. So we have two sources of carbon that can potentially be important in a marine or estuarine food web. Okay, so let's say we want to know what's the relative importance of these food sources. We go out and we collect a whole bunch of organisms and we measure their carbon isotope ratios. So here we can see that benthic worm, some fish, and to some extent some crustaceans have isotope ratios that are weighted more towards the seagrass. In fact, the worm, which is burrowing around in seagrass organic matter, looks just like seagrass almost, right? Because you are what you eat. So the crustaceans are probably eating a little bit of a mixture. They're probably moving around and they're integrating two different food sources. And then if we look on the other side of things, we have clams that are suspension feeders. They're just feeding on stuff that's in the water column. So they're not able to munch on organic matter from seagrass. They're sucking up phytoplankton and filtering it out from the water. So they look a lot like their phytoplankton. It's a very simple concept when it comes to carbon. You are what you eat. So let's talk more about isotopes in nutritional studies. I've already said this classic phrase before, but I just want to revisit trophic enrichment. So let's imagine we have an ecosystem where we have some sort of primary producer, like a benthic algae, growing on the seafloor. And this trophic enrichment, I've saved it until now because it's really effectively seen when you are looking at two isotopes simultaneously. All right, so we have a school of herbivorous fish, like these yellow tangs, that feed on that algae. And then we have a primary consumer, like this white-tip reef shark, that is going to occasionally use a yellow tang for its lunch. And we can imagine these organisms, this food web, in this isospace, this isotope space that is triangulated based on nitrogen and carbon isotopes. And as we already mentioned in the example of the tuna, with every trophic transfer, with every time an organism is consumed and digested, there is about a 3.5 per mil increase in nitrogen. This is a big change. But there's only about a 1 per mil enrichment in carbon, if at all. And those numbers are averages, and they seem to be consistent across all types of life, all different ecosystems. But there are many exceptions, and the range can be quite wide. And once again, if the shark eats the tang, then we would expect a 3.5 per mil enrichment in the shark's delta 15N, and only around a 1 per mil enrichment in the delta 13C of the shark. So this is kind of a complementary way to see how carbon and nitrogen isotopes vary within a food web. And so there are countless studies that have looked at food web dynamics using isotopes as tracers for the energy and matter within those food webs. Who is eating who? So sometimes, this is actually a very simple food web, but you can see we can have all kinds of different sources, all kinds of different organisms feeding on those sources. And what isotopes help us to do is really link the sources to their consumers. And therefore we can understand better what are the main resources that important species require to make a living in a certain habitat. Some, I think, more interesting examples are to follow. So this is a study of chimpanzees. You know, chimpanzees are pretty smart. They have a really interesting social behavior and social dynamic. And what many people don't realize is that chimpanzees are also carnivores. Well, they're mostly omnivores, but when they're capable of it, they will hunt other primates especially and eat them, other smaller monkeys, for example. And this was a study that was examining the behavior of chimpanzees in the forest, and they were tracking individuals and they were assessing their ability to hunt. And they were using isotope samples from their hair. I think they could just wait until the chimpanzees woke up and moved along and they could sample their hair, and they knew the individuals very well, that they could match the isotope ratios to the hair isotope ratio. I mean, they could match the individuals to the hair isotope ratios. Okay, so you can see they all have different names. There's Brutus, there's Macho, Ulysses, Rousseau, Darwin, Kendo, and Fitz. Okay, so these are all male chimpanzees. And the scientists identified them by their hunting prowess. So how effective were they at capturing prey and eating it? So Brutus, Ulysses, and Macho were good hunters based on their behaviorist observations. Then we had Darwin and Kendo, who were pretty poor. And Kendo wasn't really interested in eating. Darwin was an unremarkable hunter. So these guys were probably more vegetarian. And then Fitz was basically a recent adult, so he had just left his mom and started foraging on his own. So these three were unique from the hunters. And what you can see is that Darwin, Kendo, and Fitz, relative to the other chimps, had a lower isotope ratio than them, especially with respect to nitrogen. But if you look at the hunters, they're shifted up, right? And that reflects that they're eating at a higher level on the food chain, right? So if we look at that fish analogy, these guys are like the yellow tangs, right? And these guys are like the sharks. They're feeding on two different levels of the food web. And what's really, really cool is that there's an outlier here, Rousseau, who falls right into the middle. And Rousseau was identified as a cheater. So Rousseau would simply steal food from the others or eat their scraps. He wasn't playing the game very fairly. He was cheating. And looks like he was successful about 50% of the time. Right? So he was getting a little bit of protein from hunting. Okay, another, I think, exciting example comes from Africa. And this is a story that is not that old, relatively speaking, about lions, a pair of lionesses, if that's the right way to say it, that were feeding on humans. And at one point in Africa's history, there was a lot of interest in trade and building railroads. So as the railroads were being built, the people who were building the railroads would set up camp outside of the rail operations. And every night, almost, while people were sleeping, you would hear some screams, and then someone would be missing the next day, right? So then this would go on and on and on, to the point that the railroad company would hire professional hunters to come in and hunt down these lions. And what they found after they killed the lions was that one of the females had was kind of old and had some problems with its teeth, so it probably had some difficulty eating. And so the one hypothesis is that the lions shifted to eating humans because they were easy prey, even though they weren't the normal type of food that the lions would go after. And kind of creepy is that one of the lions was killed. They tracked it to its den. It it was living in some kind of cave, and inside the cave were the remains of a lot of people. A lot of people. So they found all the bones of the humans and were able to assess this rather quantitatively. And this is the figure from that paper. So this was published in a really good journal, the Proceedings of the National Academy of Science. And this is the figure from that study. So you can see the two lionesses that were shot, and the local people had the nicknames for them. One was they called it the Ghost, and the other one they called the Darkness. And because these things would come in the night, I mean, they must have been terrifying. And you can see the lions up there, FMNH23970 and 23969. Those are museum archives references, so these lions have been preserved and put into a museum. And then they compare their isotope ratios with modern lions of this part of Africa. So you can see they're quite different, actually. And they were also able to look at different parts of the lions, their fur versus their teeth, and they could look at, because the different parts of our bodies represent different times of our lives, right? Our hair is growing constantly, especially me. My hair is growing constantly. So it's reflective of my diet within the last days to months, right? But my teeth have been here for a really long time, and they're reflective of my diet probably tens of years ago. Right? And all of our nitrogen in our body is slowly turning over at a very, very slow pace. But in organs like our liver, it's happening at a much faster pace than in tissues like our muscle. So, I mean, we could go on and on about all these details, but the point is that they were able to document shifts in the diet, the recent diet of these lions, based on looking at different tissues of the animals. And then they could compare them to natural prey sources, like grazers or browsers, things like giraffes and gazelles, and things that lions normally eat. But then we have the humans, the people that were working in that environment. And what they do with this isotopic space is that they use it to calculate how much of the diet of the lions was coming from human beings. And I used to remember the number, but they actually came up with the number of individuals that they suspected these lions consumed over the course of their terrorizing reign. A really interesting paper. And actually, this is a study that was featured in Hollywood. Well, I think the study came after the movie. But this is a really good movie with Val Kilmer and Michael Douglas, The Ghost in the Darkness. It's about these lions and the hunters who were brought in to kill them. So it's a true story that has an isotope story behind it. So if you haven't seen it, it's really, really good. Okay. So how do environmental changes affect the food web? We can use isotopes to also look at the impacts of things like invasive species. If we add a species to a food web, how does it change the dynamic between different organisms? In this example, we had a lake ecosystem that is dominated by a top predator, I think it's a salmon, that feeds on zooplankton when it's a juvenile, and it also feeds on smaller fishes when it's an adult. And there's an ecosystem that's invaded by another predator, a mesopredator, that's also feeding on the fish species that the salmon like to eat. And so as a consequence of this competition, the salmon have to move into the deeper waters to get away from those competitive interactions, and their diet shifts to rely more on the zooplankton. So we can look at the impact of invasive species to see how we can change the diet of an organism. And getting into some kind of theoretical thinking of how this works. Again, we're talking about a weighted average. So an organism is a weighted average of all the sources and their amounts that are integrated from the diet. And this is an example of what's called a three-source mixing muddle. This is from a very humorous author, Brian Fry. Typically, this would be called a three-source mixing model, but he's calling it a muddle because it's confusing. So in this example, we have three potential sources of food to an organism, and they are represented by these colored squares. And you can see that they all are quite different from one another. And then we have an organism that's represented by the triangle, right? So we know that the organism is a weighted average of those sources. However, what if we don't know which sources the animal is eating? So in this case, it could be eating 50% of the yellow source plus 50% of the blue source and becoming a weighted average that looks just like the green source, but it might not be eating the green source at all. Or it could be eating just the green source and not the other two. So from this, that's why this is called a mixing muddle. We cannot resolve the trophic relationship between the organism and its food based on these data alone. So that's why nitrogen comes in very handy, because nitrogen can help us triangulate or kind of expand this relationship in a second dimension. So if we're lucky, if we look at nitrogen, then maybe that source that was unclear based on its carbon isotopes will be distinct relative to its nitrogen isotopes. And in this case, we form a nice bounding area around our organism. And we can use some simple algebra to figure out what is the relative contribution of each of those sources to the diet of that organism, if our data turn out this way. I think I've talked to at least 2 or 3 students, graduate students in ENV, who are dealing with these mixing muddles. They're really problematic. Because sometimes you can have this, where nitrogen isn't much help at all, and you basically have the same scenario. So this is unlucky. My organism is not bounded by my food sources, and therefore I could have the same conundrum. It could be a weighted average of the 2 extremes, or it could be just eating the middle food source. It's hard to tell. So there is another trick that we can do that I happen to be very fond of. We can add isotopes. We can add isotope tracers to specific sources of food in an experimental setting, and we can manipulate the food sources to our liking. So now you can see on the nitrogen isotope axis, we're going up to 100 per mil. 100 per mil. There's almost nothing on Earth that looks like 100 per mil. So I can manipulate it using chemicals that you can buy from a catalog to change that food source, and then conduct a feeding assay to see what my organism is actually eating. So this is a good transition into talking about the last section of the lecture, which is about isotope tracers. And I say this is when things get wicked cool, because isotope tracers can be utilized to manipulate the biology, to amplify the biological processes that are active within living organisms. And I'll give you a few examples from my own research. So we often use stable isotopes as metabolic tracers. We're looking at the metabolism of organisms. Things like digestion, respiration, photosynthesis. These are all metabolic activities. In corals, we have a whole diversity of metabolisms because we have an animal, the coral animal, which is just like a jellyfish, that has evolved a relationship with a plant, these dinoflagellate cells that live inside the animal cells. So this is a very intimate relationship, and through this partnership, the coral has a toolkit of metabolisms that almost no other organism on the planet has. It can eat stuff from the water column. It can photosynthesize, just like a plant. It can generate amino acids that are taken up from inorganic nitrogen sources, just like a plant. Or it can derive nitrogen from feeding and assimilating proteins from a whole variety of food sources in the water column. So in order to make sense of all this, we have to utilize these isotope tracers to amplify what it is they're actually doing. And I'll give you an example of how this works. So no one's done this before, and I haven't yet published any of this stuff, but we've been playing around with deuterium. Remember, this is the heavy form of hydrogen. And deuterium is cool because we think it's a good tracer for photosynthetic processes. In the ocean, the biggest source of deuterium is ocean water. So you would expect that animals growing in ocean water would have the same isotope ratio as ocean water, which we set as 0. But corals are not 0 at all. And in fact, when we started this incubation, the corals had an isotope ratio of minus 140 per mil. So this is a natural abundance isotope ratio, and this is reflective of the fact that any fractionation against that heavy isotope of hydrogen, which is 100% heavier than the normal isotope of hydrogen, is going to result in a huge, huge difference, right? So that huge mass difference is reflected in this very negative value. And when I add deuterium, when I add the heavy hydrogen to the water that these corals are growing in, as they photosynthesize, they're incorporating that heavy deuterium into their bodies. So you can see the isotope ratios go up in the daytime, and then we turn off the lights, and they go back down. So they're losing that hydrogen back to the water. And then you turn on the lights again, and up and away we go. So we can use deuterium to track these photosynthetic processes. And so that's happening inside the coral's symbiont, and we can look at the host, the coral animal, and we can see that it's benefiting from the products of that photosynthesis. It's tracking its symbiont very closely. So that's a rare example of using deuterium. This is carbon isotopes in corals. So we can use carbon isotopes to see how much carbon is being assimilated to the coral through photosynthesis. So here is the natural abundance isotope ratio, almost minus 20 per mil. And if we incubate the coral in the presence of our stable isotope tracers, it doesn't have a higher number. It's a pretty low number still. Because in the dark, we can't assimilate any of that isotope tracer, which is in the form of labeled bicarbonate. But in the light, when we turn on the lights in the presence of the tracer, we're now getting an isotope ratio of over 500 per mil. Over 500 per mil. So we've just really, boom, we can quantify now how much carbon has actually been taken up by the coral and fixed into organic compounds. And if we look at the host animal, we can see that they're tracking their partners very closely. So the host is very clearly benefiting from their symbiont's photosynthesis. So this is a very powerful tool. The next example is not the best example because we're looking at ammonium. Ammonium is a very bioavailable form of nitrogen. And we can add labeled ammonium to the media to see how the ammonium is incorporated into these organisms. So we know that the symbionts are the only cells that have the ability to assimilate nitrogen from ammonium. And when we add ammonium to the environment, whether it's light or whether it's dark, they're taking up some of that signal. It's just that these data aren't the strongest data that I have, but you can see some significant enrichment. And more importantly, the nitrogen that's being assimilated through the symbiont is accumulating in the host. So the host appears to be getting the products of that ammonium assimilation in the form of amino acids. So again, this is a really powerful way of resolving the biology of these organisms. Okay, so that's enough about isotope tracers. And I just want to talk, I think I have one slide on how these instruments work. To measure the stable isotopes of a material, you have to use a mass spectrometer. And traditionally, a mass spectrometer is used to measure the mass to charge ratio of a molecule. It's informative for identifying molecules. Okay, that's traditional mass spec work. But an isotope ratio mass spectrometer is looking at very specific elements, very specific compounds and it's able to detect the variation in those compounds based on one atomic mass unit's difference. So think of this as an extremely high precision mass spectrometer. These types of instruments are not meant to look at all compounds, only very specific ones with very high precision. And the anatomy of a mass spectrometer is something like this. You have a source, and a source is, consider it a light bulb. It's kind of like a light bulb. In fact, it even has a little filament inside. And that filament, through that filament runs an extremely high current. So, these lights might have a few volts running through them. But in an isotope ratio mass spec, we have 8,000 volts, 8 kilovolts, running through the filament. So an extremely high amount of electricity that provides energy for ionizing compounds, ionizing elements that enter into the source. So we introduce a gas that we want to measure into that source, and it becomes highly energized, highly ionized. And because the whole system is kept under a vacuum, those ions accelerate down a flight tube, which is represented by this arc, this arcing shape. They're flying down this flight tube all bound together. So all of the isotopes are accelerated down through this flight tube, and then they run into a magnetic field. And the magnetic field works something like this. Imagine you have a bowling ball, and you have a baseball. And you throw the baseball, and it goes up in the air and comes down somewhere. And then you take the bowling ball, and with the same amount of energy, you throw the bowling ball up in the air, right? It's going to land much closer to me. So the difference in the trajectory of those two objects thrown with the same amount of energy is different because of their mass. So as our ion beam is traveling and encountering this magnet, the magnet exerts a force on those individual ions. And the heavier ions are going to actually have more inertia, so they're going to travel a little bit further. And the lighter ions are going to be pushed very easily by the magnet, and so they're going to take a tighter trajectory, a tighter arc. So as a consequence, when we have originally one ion beam with all of the isotopes inside of it, we can get multiple ion beams with only one isotope represented by each. Okay? And then because we have these systems designed for measuring very specific things, we have these little tiny targets, these little collectors that are waiting for those beams to strike them. And every time an ion makes contact and discharges on that target, a signal is sent to a computer that, hey, we have a heavy carbon atom here. We have a 13C. We have a 12C. And it's literally counting every single impact. And that's how it works. It's actually a pretty simple technology that's been around for quite some time. And at Hong Kong U, we're fortunate to have a stable isotope lab that I hope to, if I don't get to take some of you into the lab, I hope to do a video tutorial so you can see what it looks like on the inside. Unfortunately, it's a very small room, so we can't have a class in there. But in our laboratory, we have what is known as an EA. An EA is an elemental analyzer. An elemental analyzer is used for measuring the carbon and nitrogen content of organic material. So a lot of people who have labs that are just doing soil analyses or plant analyses, they would have a standalone elemental analyzer all by itself. What it does is it takes a sample and it burns it, combusts it at high temperature, and then it can separate the CO2, it converts the combusted gas into carbon dioxide and N2 gas using some chemical reagents. And then it separates the CO2 and the N2 gas on this long column that slows down the flow of CO2 and allows the N2 to go through very quickly. So you get a separation of the two gases in space and time. And then as the gases come out as a single pulse, it can measure the magnitude, how much of that gas came out of the sample. And if you knew how much sample you put in there to begin with, the instrument will tell you the percentage of your sample that was carbon or nitrogen. So by itself, an EA is a very handy tool. But what's really cool is that you can plug an EA into an IRMS. And what's happening is you're using the EA's combustion capabilities to convert an organic sample into CO2 or N2 gas, and then you're sending that material straight into the mass spectrometer for isotope determination. It's a pretty cool system. And we now have, actually they're working on it right now, we're adding what's called a high temperature EA, HTEA, or we can also call it a TCEA, thermal conversion elemental analyzer. Instead of using combustion, as you know, we use a little bit of oxygen and high temperature to burn something. Instead of using that, we're using pyrolysis at about 1400 degrees. It's the thermal ripping apart of atoms. So if you add a little bit of water into a pyrolysis furnace, you're going to generate hydrogen and oxygen gas, very, very high temperature at 1400 degrees Celsius. So if we add a piece of my hair or your fingernails to this TCEA or HTEA, we can determine the hydrogen and oxygen isotope composition, which is reflective of the water that you drink, and to a lesser extent, the diet that you eat. So in forensics, isotopes are hydrogen and oxygen isotopes have been applied to the hairs of people who have been maybe they've been found far away from their home, they've been kidnapped or murdered, unfortunately. People can use in forensic investigations the hair of the person that was produced right before their death to trace the isotope signature of the water that they had been drinking. And it just so happens that water isotopes vary quite predictably across the continents. So you can use the isotope data to determine where that person had been prior to their demise. Okay, so these are very powerful tools. And with this device, we are capable of measuring in bulk organic material nitrogen isotopes, carbon, oxygen, and hydrogen. And we will have the ability to look at water, to measure the isotope ratios of water and how that varies in different environments. We also have a really cool system called a GC IRMS. So a GC is a gas chromatograph. And I hope in the future, probably not this year, but we will incorporate some gas chromatography into this course. But gas chromatography is when you have a liquid, a volatile liquid sample that's injected into this instrument that basically heats it up and turns it into a very volatile gas, right? So the gas is sent through a very long, sometimes 60 to 100 meters of very thin capillary tubing that's coated on the inside with a special material that, just like in our EA, is sticky. It's sticky to some compounds and it lets other compounds go more quickly. So depending on the polarity of the compound, the size of the individual compounds, they all get separated in space and in time. So they'll come out one at a time and they go through a combustion interface where they get burned one at a time, turned into CO2 and N2 gas, and then they go into the mass spectrometer. So what does this allow us to do? It allows us to measure the isotope ratios of specific compounds. And here at Hong Kong U, we can measure the carbon and the hydrogen isotopes of specific compounds, so amino acids, fatty acids, and hydrocarbons. So think of it this way. All of the data I've shown you today are from EA IRMS. And every data point I showed you is from one sample. You put one sample in, you get one carbon value, one nitrogen value. But when you do something like the compound-specific analysis of an amino acid, I can take one sample, I convert it into all of its component amino acids, and I can measure all of them in one analysis. So I can get maybe 13 data points if I'm lucky. So this is I would say that this area is the cutting edge of isotope science right now, isotope ecology. Okay, so this ends the lecture part of today's class. And as you can see, we have a little bit of time left. And I want to use that time to talk about something that you hopefully are thinking about, your group projects. So what should you be doing now? Does anyone know? Well, we put in the syllabus an assessment, which is a outline of the report that you're going to execute. And I bumped the deadline back to Friday because I didn't think anyone would be ready to hand it in today. What you should be doing, if you haven't done so already, everyone has handed in 5 papers based on their project that they're selecting. You should have read those papers to the point that you understand them. And then you should look for more papers, right? 5 papers is not a really representative sample. It's not enough for you to really understand the topic that you're going to study. So you need to find more papers. There's no limit to the number of papers that you should look for, but look at any scientific publication. Typically, they have something on the order of 30 to 50 scientific papers that are cited. Okay? I'm not going to give you a magic number. You just need to find the references that will help you get the job done. You need to work as a team to read and understand the papers. It's not enough to just find the papers that have the closest matching headlines, okay? The closest matching titles. You actually have to dig into the papers and learn something from them. Now, every paper you find, you may not need to read it word for word. You might only read the abstract, and that might have all the information you need. Or you might say, okay, I just need to know how they did this. So you go straight to the methods section and you read the methods section. Okay? Don't think of this as like you're reading a novel. You're not reading a novel. You are looking for and collecting information. So you need to do that quickly and efficiently. If you're totally lost and confused and you don't even understand the basic concepts behind the paper you're reading, then maybe you should spend some time with the paper and maybe Wikipedia and try to sort things out. And if it's really complicated, let's talk about it together. Okay? So this is not a solo activity. You have a group, you work together, you divide the labor, you read the papers, you share them with one another. I learned this from that paper, you learned this from that paper, and so on. So that's how this should work. Read the papers to understand them, and share the information with your team. Some of you have already met with me or some of the demonstrators to talk about your projects. If a group has not done that yet, I would urge you to set up something today or this week. I think we're going to have time to hang out today and have some group meetings. And you should begin thinking about framing the work that you're going to do. I know you haven't done anything yet, but you should start thinking about the basics. What is the problem? What is the question? What is the environmental problem that I want to address with my research? Some of that information you can find in the list of project descriptions, but it may not be exactly what you want to do, so you can feel free to adapt it. And it shouldn't be just a cut and paste kind of thing. You should delve into what I've given you and expand upon it. And this will become the introduction, the very important introduction part of your report. Okay? So all the while, even though maybe we haven't gotten to the methods that you're going to be using in the class yet, or maybe you're still unfamiliar with some of the methodologies. There's no reason that you should just wait until you get that information before you start thinking about the environmental problem that you are challenged with learning something about. Is that clear to everybody? So please don't squander your time. Unlike some of your other classes, there are not as many regular assessments in this course. But when it comes to the end of the class, you are all going to be swamped because you've put off everything until the end. So now is my message to you. Put some effort into this now. Get started reading, writing, thinking. And then once we get your data, everything is going to come together very quickly. Okay? So I urge you to start thinking about this stuff. And even begin writing. And you also have to think about your plan. What do you need to do? Some of you are going to do something in the lab. It could take a day. Some of you are going to require sampling that requires a few weeks of regular visits to a site. So we need to start talking about how are you going to execute your project? What is the time frame for completing that plan? And will you be able to have your data by the time your report is due, most importantly? Okay? So that's the type of thing that we can have a discussion about. All right, so with that,